Good morning, everyone. My name is Daiki Nomura from the Hokkaido University, one of the chair of this session. So our session was from morning and afternoon, and poster session in the evening, maybe. So today is a big day for our session. And yes, in this morning session we have a special talk. So please.
Thank you very much. Yes, this is pretty special indeed. It's really nice for the Geochemical Society. I'm Barbara Sherwood-Lawler. I'm the past president of the Geochemical Society. And along with current president, Lord Weisberg, we're here today because of a new initiative in the Geochemical Society. Thanks to an anonymous donor, it's been a donation of the funds to establish in perpetuity and a now endowed lecture in biogeochemistry that will take place at every Goldschmidt, but beginning with this one, the inaugural lecture in biogeochemistry. It was a nomination that went out to the community several months ago. It was a competition in which we had 5 different nominees, and then an independent committee struck to take a look at the nominations and select this year's lecture. It was an incredibly strong field of nominations. So it was, in some ways, it was a difficult choice because all the nominees were great, but in some ways a very, very easy choice. Because I think many of us here feel that it is just so entirely appropriate. And it's certainly a great, great pleasure for me to have a small moment to introduce this year's inaugural endowed lecturer in biogeochemistry, Dr. Antje Goldschmidt. And of course, Antje is a professor of geomicrobiology at the University of Bremen, but also the leader of the Helmholtz-Max Planck Institute there and research group on deep sea ecology and technology. It's extremely difficult to introduce Antje because one has to select from the host of awards and recognitions that she's held. Most recently, for instance, election as a fellow of the American Geophysical Union, election as a fellow of the American Academy of Microbiology. It just continues. One of the important points to mention, though, is a leader not only in science research, but also in science policy and advocacy, both at the European level, the highest level in Europe, but also internationally. A real voice for science and geochemistry and geomicrobiology worldwide, for which we're all grateful. And most importantly, of course, chair of our 2017 Goldschmidt in Paris next year. And without further ado, thank you, Antje, and congratulations.
Thank you. Thank you very much.
I'm very pleased. Thanks so much for nominating me and for also awarding me with this endowed biogeochemistry lecture. It says something really nice. It is for those people who do brave field work, who are innovative and do cutting edge work. And this is, of course, to me a great pleasure, because where else can you do cutting edge, exciting, innovative work than in the Arctic, where you go to these extreme environments, where you live in the ice, on the ice, where you spend months in the dark to find out what the ice holds in terms of life. And this is the story of today. I would like to talk about the declining sea ice cover of the Arctic Ocean and about the biogeochemical effects this has. And I would like to focus on how life matters in all of this. So sometimes we biogeochemists think we just need to compute the right numbers and then we understand, but then we encounter the diversity of life and the special adaptation life has, and we see that it is really in the end about life to determine biogeochemical fluxes. And I would like to start with my great hero in innovative cutting-edge field work in the Arctic. That's Fridtjof Nansen, sitting here in front of his ship, where he got frozen in his famous farm expedition some more than 120 years ago. And he was really thinking a lot about the Arctic and about the right way of doing science in the Arctic. At his time, it was about finding out whether there was a continent, a hidden continent in the Arctic, whether it was truly an ocean or whether there was a secret island, how deep the ocean was, how cold it was. All of this wasn't figured out 120 years ago. But he was also very concerned with life, and I can recommend to everyone interested in the Arctic reading his books. He published a lot about his adventures in front of the microscope. So despite he was such an adventurer, for him the most beautiful thing was to sit in the ice with his microscope. His colleagues thought he's lost from science and adventure because he could stare for 12 hours a day onto what the little life he saw under the microscope. And he described it very carefully, and I love how he says, Mother Nature has an amazing ability to generate life everywhere. For her, even the permanent ice is fertile grounds. Little did he know at this time that the Arctic sea ice isn't that permanent at all. So today, we are looking with awe at the change of our planet and the change of the Arctic. This is the compilation of the temperature anomaly we see from 2005 to 2014. And in the Arctic, we are already in an above 2-degree world. If you have followed what has happened last winter, we had jumps in temperature of plus 30 degrees from an average minus 25 to what our buoys up there recorded in wintertime, sometimes 5-degree plus in the Arctic. So this is a part of the Earth where the change, we can really say so as scientists, is dramatic. And I love this little clip put together by NASA to make us understand how rapid this change is and how unforeseeable it was. You're looking at the sea ice cover as seen from the satellite. This big ice-covered or glacial-covered island is Greenland. And so here you see what happens. In 80, we had 30 years of the CO2, the Keeling curve. In 90, IPCC started getting strong, and still we didn't foresee this decline in sea ice. Towards 2000 was when Arctic researchers said, there is something happening. And in 2007 was this record low, right in time for the polar year that was prolonged then. In 2012, and this is what I'm going to discuss now, the second low. And not only did we lose that much sea ice cover, as you have just seen, but we also lost the multi-year ice for great parts. So when I was a PhD student studying the sea ice, it was at this time in 93 where we would have a multi-year ice cover in the Arctic Ocean where the average thickness was some 4 to 5 meters. And today we have lost this thick ice and we have retained the 1 meter thick ice or less than 1 meter ice. Of course, in wintertime it all freezes back, but even in wintertime we see a decline. So this is what we call the Arctic amplification, the big Arctic change that our models cannot reproduce. And the most important bit is actually understanding what it looks like. Here you are now in the middle of the Arctic Ocean, close to the North Pole. In 2012, you look at the ice from the helicopter flying above Polarstern, and you can see what has dramatically changed from Nansen's time. The melt ponds, the melting of ice on this 1 meter thick ice has completely changed the picture of the landscape. We have increased by an estimated 40%. Unfortunately, satellites cannot count or measure melt ponds yet. We hope that future missions can do that. And it doesn't end here. This is the actual curve of the sea ice satellite, what it sees today in 2016, and you can see we will hit a new record low this year. You can predict what comes out already March, April, and basically we will undercut 2012, and we don't know where the end of this is. So how does biogeochemistry change if the one defining characteristic of the Arctic changes, and this is the sea ice? First of all, of course, and this is hard field work, when we go out there, we have to understand light. So we all know that photosynthesis, which starts the biogeochemical the carbon cycle, depends on light. The thick old sea ice of the former Arctic, with 4 meters thickness and some snow on top, has reduced light availability to plankton for a short season at a time. And today, with this emerging huge melt pond landscapes, we get almost an order of magnitude more light, but only for 1 or 2 months. Understand that, of course, the availability of sunlight depends on the Earth and the Earth's angle, Earth rotation, Earth angle, and this will not change by global change. But if you remove ice and snow cover, of course, there's much more light available for under ice algae growth. So the sea ice physicists of AVI have done these field measurements, and they have tried to upscale the thinning ice and the new light availability. And what they can tell us is, especially here you see again the picture of the Arctic. This is Greenland for orientation. These are the large Siberian seas. And I would just like, before we look at the light availability, like to point out to you how special the Arctic Ocean is. This, you see a bit of bathymetry under the ice cover here. And note, these are the vast shelves of the Arctic Ocean. It is a giant shelf sea on the one hand, but then it has these very deep basins with a depth of 4,000 to 5,000 meters under the ice. So in 2012, the ice retreated to basically depths of beyond 1,000 meters. The shelves were not ice covered. This is the probably new northern sea route that some ships are willing to sail in the future. And this is light transmission. And so you see that in here in the Siberian seas, we have much higher light availability for plankton, even in and under the ice. And here we're kind of stuck. Our ability to see ocean color and estimate photosynthesis is limited by the ice. The satellites cannot predict photosynthesis or ocean color when there is ice in the way. So we have nothing but our field measurements. And the big problem that Paul Wassmann and his team from the University of Tromsø always point out is this is the map, again, with the average ice cover that we now constantly undercut. And these are the long-term measurements that us people have for understanding change. And you can see that basically the long-term ecosystem time series, and these are all the international ones from everyone, they basically are not in the deep basins, and they are not in the area that changes the most. And for me, this is also very sad. I believe in presence at sea and in the ice for understanding what changes, what the changes will have in terms of effects on biogeochemistry and life. So what we are now going to discuss in the next minutes is basically seasonality and this one chance for phytoplankton to grow when there is enough light, when the ice melts in summertime, which is a natural process. You can see that by this depiction that there is like this one chance for about 2 months, June, well, to some part in August, for phytoplankton to grow and for all the grazers to eat it, for the zooplankton then to have enough lipid stored to overcome the long winter and to breed again. So Arctic organisms are highly adapted to this strange environment. There's just this one short time for light, for growth, for reproduction, and all has to happen in this time. And any changes that occur, for example, when the phytoplankton bloom occurs, will affect these inner clocks these organisms have. So that's a big question that we biologists and biogeochemists have. How will basically the food web adapt to these changes of a much earlier spring bloom? And what will be the role of the ice algae in the future? In 2012, we didn't foresee this great sea ice minimum, but we were out there with Polarstern. There was also the Chinese icebreaker out there doing measurements, so we had at least 2 ships making observations as well as autonomous sea ice buoys. And so I will focus first on 2 areas. First, I'll start with the Laptev Sea shelf. Here you see the Laptev Sea shelf. It's part of the Siberian seas. Again, when I was a PhD student, it was fully ice covered. In summer 2012, for the first time, fully ice-free. And this is the shelf, and here you go from about 200 meters down to 4,000 meters. So this is the first area I'm going to focus on. Then I'm going to focus on the ice edge and the inner basins, and the talk after me by Ralf Hoffmann will focus on Fram Strait to give you another picture of changing sea ice in Europe then. So what did our fieldwork consist of? Basically, we used Polarstern as a platform, went into the ice. We got ourselves partially frozen in from time to time and drifted with the ice to do our observations. And these included the water column. They included basically the sampling of the ice and under ice and the melt ponds. And we also brought a new component to our measurements, which was the benthic observation. For the benthic observation, we used lander systems. These are systems you launch from the ship. They sit on the seafloor and they do microprofiling and chamber measurements of the seafloor activity of microbes, respiration rates, for example, nutrient fluxes. And we use a camera sledge to survey the seafloor at the same time. For us, the seafloor is the ultimate sediment trap. So where sediment traps may have problems in being quantitative, the seafloor is, of course, quantitative. We just have to get to it. Let's start with hypothesis number 1. For example, Kevin Arrigo and his team brought forward the hypothesis, if we decrease the ice cover, here you see the ice, the satellite cannot see through it. If we increase light availability, then we must have an increase in photosynthesis. And he predicted for the Siberian shelves an increase by about 20% maybe. So note this is from 450 to 600. And we could test this at our times, basically because I stored my PhD samples, and I always thought I come back one day. I took them in '93. when there was ice cover, as you can see, and I thought one day I'd come back and look again into the samples and into this area of looking for change. So this we had, and please note the orientation. So this is the shelf, this is the deep sea, northwards, and here it's turned around. This is northwards towards the North Pole, here the shelf, here the deep sea. So you can see along the slope, what you see here is photosynthetic pigments, chlorophyll at the seafloor, a good tracer for carbon flux by algae. You see that it decreases with water depth, but there was quite some pigments to measure. And we could see that, yes, when you have more light on the Siberian shelves, going from about 200 meters, the outer shelf edge, down the continental slope, we have already increased in the past few years the amounts of pigments falling to the seafloor at all water depths by, in some areas on the shelves a little bit, but mostly by 200% along the shelf. So more than is predicted by the satellite view. We have a large drawdown of carbon onto the shelves to quantities that were probably not seen before. And now it's very interesting to see if this will continue in the future. And how did the seafloor respond? This was what we also looked at. So Christina Bienenhold here did lots of measurements along the slope to find out how the seafloor responded. So what we did, for example, we measured the change in microbial activity, and what we measured is a bulk community level measurements of hydrolytic enzymes. It's basically digestive enzymes, the same enzymes that we have in our gut. If we eat some bread or so, we make some alpha-glucosidase to degrade it. So we looked for those enzymes that degrade cellulose components as brought down by phytoplankton, and we looked down for enzymes that degrade what is brought down by zooplankton, carcasses, fecal pellets, and so on. And you can see that both for phytoplankton hydrolytic degradation activity and for fecal pellet zooplankton remnant, that microbes increase their activity, their digestive activity. They can digest more, and especially when it came to chitin-containing food, similar to what we saw in pigment increase, we saw in enzymatic activity. And also, we compared, did the communities change yet? So we measured by molecular methods, and I will not go into detail here, if the communities that we sampled along the way from the shallow shelf areas to the deep sea, did it change between 93 and 2012. The dots with the stars are the communities sampled in 2012, and you can see that there is maybe a tiny shift, but it cannot be resolved against water depth. So basically we have this strong grouping of microbial communities according to water depth, and it still seems more or less the same community, and we confirmed this today by high throughput sequencing that yes, we are looking at the same microbes. There is no big change yet. If we record a change, it's more in the eukaryote area. And we have basically teamed up with the Russian zoologists who did sampling of the macrofauna. What we can see today is there is a change in the benthic community starting from the macrofauna, basically in those areas that received a lot of the unusual phytodetritus fall. We have the first increase in polychaetes, showing that there will be more, that there is more food, probably more recycling at the seafloor in the future, leading also to changed microbial activities and eventually a new benthic food web. Now, when you know the Arctic, you know, of course, that light is not the only thing that will change. The Arctic, when it warms, will be a highly stratified ocean, even more so than today. Today, it has some, in summertime, some mixed layer of maybe 25 to 30 meters. The more warming you have in the surface and the less ice cooling you have, the more stratified the ocean will be. And in the Arctic, you have very little nutrients. So we know from the records that we have that the Arctic is nitrate limited, most of all. In some areas also phosphorus and silicate limited, but not so much in the Siberian margins. So hypothesis number 2 to be tested, this growth of phytoplankton cannot be for very long. They will hit the nutrient limitation very soon. Is there any organism who can adapt and who could be the winner of the situation that we have in the Arctic? This is the extra question that we wanted to answer. Now let's have a look for a moment into how you can figure this out for the Arctic Ocean. It's basically a great reactor by this strong stratification. What you see here is the temperature curve of the seawater. In wintertime, the top Well, depending on where you are and how fresh the seawater is already on top, you'll have maybe some 50 to 100 meters, in some areas in the central basins 200 meters that will cool, where nutrients will mix, but you have very little nutrients to start out with. The spring nitrate concentration in the Arctic surface waters under the ice, where no productivity happened during winter time, has been some 8 micromole. We don't know what it is today. In the Siberian seas, it is that low. When you go to the Canadian side, it is higher, but we are talking here about Siberian seas. So you start out productivity with some 8 micromole, and then it starts warming, and then you have only some 20 meters before you go to basically the thermocline. So some 10 to 20 meters of 8 micromole doesn't carry you very far. You can measure what is consumed in terms of nitrate because there is the winter haline zone where you have the maximum convection, and basically if you know what nitrate you had in the surface and at depth, you can figure out what was consumed, and you can calculate nitrate consumption, and by Redfield ratio you know photosynthesis and carbon fixation. This is a difficult measure to do. For those of you who have looked at autonomous buoys, there's quite some zigzag. We have several thermoclines and also the ongoing freshening. So in some areas, the mixing layer has been reduced to less than 10 meters. Here you see what Polarstern sees when we equip it with nutrient sensors and nutrient measurement capacities when it sails these areas. It is new that we can do that with Polarstern, that it is a fully automated nutrient lab. And please look at the colors of nitrate. This is in July and August, and when you go into the ice, and when you go into the central Arctic basin, so away from the shelves that are influenced by the continents and the rivers, you actually have 0 nitrate in basically all of the upper mixed layer. That means you reduce you consume all of the nitrate, you have some phosphorus and loads of silicate left, so there is no limitation in phosphorus and silicate in the Siberian central basin, but you run out of nitrate quite early. We don't know how early today. There are no mixing processes. This is melted fresh water on top that warms by the atmosphere and by warming through sea ice even, and there are no winds in the ice. Only in the open areas we can hope for some winds. And what to me is the biggest question of the future, will we have more nitrification? Will we have higher ammonium fluxes, nitrate fluxes from the shelf seas to replenish the nitrate? Because also the inflow areas will not carry more nitrate, because here it has warmed already and the spring bloom is earlier both in the Atlantic and in the Pacific area. So we foresee, this is our hypothesis, that the Arctic will actually be totally depleted of nitrate, and that the big winner for this period right now is a funky sea ice diatom called Melosira arctica, which is known since quite a while. It has this shape, it forms long chains, it makes these gigantic carpets. It almost looks like kelps hanging down from the ice for 8 meters or more. Here you see a brave Russian diver taking samples from these carpets. And this algae is sailing with the sea ice through the nutrient-depleted Arctic Ocean. It uses the sea ice as a bus. It is carried around. It can pick up slow amounts of nitrate. It concentrates it against the gradient, stuffs itself with nitrate, and has been named the algae of the year 2016 for its great adaptations to a changing Arctic. Let's visit it for a short moment. And then you can see why we need presence at sea. The satellite cannot see any of this. We cannot see any of this until we dive under the ice and see these algae growing. And even in a fully nitrate-depleted environment, they are growing in the ice. So now you wonder, how can a diatom do this? It has no roots. It has actually, how does it make these kelp-like beds under the ice? It's a fantastic algae, and Mar Fernandez-Mendez, now at the University of Tromsø, did some cool experiments with it. Basically, it produces huge amounts of slime. It sticks its chains together, makes these long filaments, and the slime freezes into the ice when there is freezing processes, so it holds itself onto the ice. As I said, it can pick up nitrate, concentrate it to high quantities inside of the algae. It forms bubbles to float. So when you are mean and torture these algal communities a bit and you poke with a needle and kill the oxygen bubbles, it gets too heavy for its thick silicate walls and sinks to the bottom. When you then give nutrient again and some more light, it will float up again and freeze back into the ice. So it's community behavior that can completely outcompete the harsh environmental conditions around. And the greatest adaptation of this algae is it must taste horrible. It's not eaten by any critter that we know. You see hungry amphipods swimming by and they can't just not eat Melosira. So Ma found out that this algae in 2012 was the winner. It made most of the primary production and most of the export flux, as you will see in a moment. But what was magic for us benthic biologists, when we put our cameras down, and this is under the North Pole, we saw the seafloor littered with these greeny, slimy algal mats. So what we found is that Melosira, when it's not happy, when it cannot do photosynthesis, eventually when it gets too nutrient limited, or actually when it gets too warm, it's very sensitive against temperature. There have been old observations off of Greenland that it sinks by above, when the water gets above 1 or 2 degrees, it's unhappy. It lets go because its evolutionary ancestor is a benthic diatom. It has one of these old, well-known diatom cycles that if you're not happy, you better bury yourself in the sediments. There will be storms and eventually you come back. Of course, not in the central Arctic basin, but at least here. We could see they fall to the seafloor everywhere. And when we then poke our microsensors inside of these mats, we can see that the microbes respire this carbon, of course. So we had the first anoxic patches in the Arctic Ocean probably since, well, maybe 1 million years. So this is new, as we think, because when you go deep under into the sediments, you actually see that the oxygen is higher, deeper under the patches, saying that there is fresh consumption on top. So we calculated a large downfall of carbon for the summer of 2012. And now we got our sediment traps back finally, and we evaluated them, and we caught the same picture in the sediment traps. You can imagine it's really hard to have sediment traps under the ice and get them back. Here you see how different, there is no sedimentation in winter, almost none, but in summer there is a lot of algal fall. And when we looked inside of the trap, this is work by Efi Nötig and Catherine Lalonde from the AVI. Here looking at the surface traps, we saw the same picture. There is always fluxes in July, August, September, very little in spring and summertime. They found the usual Arctic diatoms in summertime, but these big fluxes were already made up by Melosira in the surface trap. When they looked at the bottom trap, they actually saw no more common Arctic algae making it down to the seafloor, but huge pulses of Melosira arctica, especially with loads of resting spores that they sediment to the seafloor. So basically, what we got is a huge input of Melosira, huge growth of Melosira in the Central Arctic. We think this is new, although even Nansen has reported of Melosira from growing under melt ponds. These kinds of mats that we are seeing today, we think is an adaptation to the changing Arctic. And when they are not happy, when it gets too warm, they fall. And you can imagine that they not only take down carbon, but they take nitrate down big time. Because they are full with it, they are concentrating nitrate and they take all of this down. So here I come to an end of this story as to how life matters in a changing Arctic Ocean. What we predict is going to happen is that the increasing stratification will be a problem for the common Arctic diatoms that have not these means of adapting and riding in the sea ice as with the bus. The polar mixed layer will decline and decline, and please look at the autonomous buoys that we have out there with international efforts, especially Japan is doing a great job in putting autonomous buoys to the Arctic. But we have this strange diatom algae that originally comes from a benthic diatom and has found a ways to adapt to this environment, but it will run the Arctic fully out of nutrients. So our model estimates is that in a few years, we cannot foresee any way to put nitrate back into the Arctic, except the large shelf seas will probably pick up in remineralization and send back some nitrate. So where are we today with understanding the changing Arctic? And we have this great session today and the poster sessions to discuss much more. Some people are extremely worried, and it's no secret that I belong to them, that I foresee that we have a huge change not only in the habitats, the endemic species, what the Arctic Ocean is. We know today that our extreme weathers come from changes in sea ice cover, so us humans will feel all of this. We change the culture, there will be traffic, there will maybe be gas and oil exploitation, and so on. For some people, this means those areas where in Siberia, in Northern Europe, in Alaska, you can foresee maybe for a brighter future. It will be warmer, you may be able to grow wine up in Norway. So we humans have to decide what the world should look like that we live in. It is important to talk about it, and I make some advertisement here for the Arctic Frontiers in Tromsø, where all the stakeholders meet and discuss. We fight, we discuss, we show our data, no matter if you are a boss of an oil company, if you're the Prince of Monaco, or if it's our scientists, that's where you meet and discuss how we are looking into the future of the Arctic. But I can tell you one thing for now. Fridtjof Nansen was very worried about the ability of science to be out at sea, to be present, to do the right measurements. He was the one inventing the first thermometers that were calibrated, and he wrote a big paper in 1906 about science failing if there's not the most accurate measurements out there. How can you do that? We need, as biogeochemists, to come together and be as accurate as the physical oceanographers are. We need to calibrate our nutrients. When we tried to find out the status of nitrate, did it increase in the basins already? We noticed that our international ability to do these measurements is limited by ±1 micromole, and it matters to resolve the changes that we have. So my proposition to you is, let's think more about it. Let's try with international efforts to be out there in the Arctic. It's the greatest experiments humans do with the planet today. We need to be there. We need to be present at sea and in the ice, and at least report to future generations, like Nansen did to us, how was the Arctic like before the Great Warming. Thanks for your attention.